Have you ever, found yourself standing in the supplement aisle, just staring at this massive wall of probiotics?
Oh, it is completely overwhelming. There are so many options.
Right, and you're probably wondering why a specific bottle seems to work absolute miracles for one person, but, does absolutely nothing for you.
Yeah, you're swallowing billions of these little bacteria, just hoping they'll set up a thriving community in your digestive tract.
Exactly. But for some reason, certain bacteria manage to anchor themselves and colonize, while others, well, they just pass right through your system completely undetected.
Which is incredibly frustrating when you're paying for them.
Tell me about it. So today we are taking a deep dive into the underlying mechanics of exactly why that happens. We're unpacking a fascinating 2026 paper published in the journal Cell Genomics.
Yes, by Wang and colleagues. It's a great paper.
It really is. It's titled Bacteriocin-mediated Ecological Adaptation. And our mission for this deep dive is to figure out exactly how one specific strain of bacteria manages to set up camp in the incredibly competitive, frankly cutthroat environment of the human gut.
You really do have to view the adult human gut as a fiercely guarded metropolis. I mean, it's not an empty landscape waiting to be settled.
To prime real estate, just sitting there, right.
Exactly. It is a densely populated, highly organized, microscopic city. Every single micrometer is taken, and every nutrient is fought over.
So it's basically a microscopic turf war.
A vicious one. And we're going to explore the specific genetic mechanism, literally down to a single, highly specialized gene that allows one bacteria to infiltrate this city, fight for its space, and systematically reshape its entire neighborhood.
I love that. So our main subject in this turf war is a bacterium called Bifidobacterium breve. Now, if you follow microbiome science, you know B. breve is famous.
Oh, absolutely, a superstar microbe.
But it's usually famous for dominating the infant gut, right? It's one of the pioneer settlers in a baby's microbiome.
That's right, which is what makes this study so interesting. The researchers started with this glaring anomaly. They found some rare, adult-derived strains of B. breve.
Meaning strains that somehow managed to thrive in the fully developed, highly crowded adult gut.
Exactly, which completely goes against the standard ecological models we've relied on for years.
Right, because an adult gut is so different from a baby's gut. So the central question the research team had to answer was simply, what makes these adult strains different from the infant strains?
And their initial hypothesis was grounded in, well, basic metabolic logic. Infants and adults consume radically different diets.
I mean, hopefully, right.
Right. Infants rely on breast milk or formula, which are rich in human milk oligosaccharides. But adults consume complex, plant-derived polysaccharides.
Lots of fiber, complex carbs, that sort of thing.
Exactly. So the researchers hypothesized that the adult strains of B. breve must have evolved a different genetic toolkit just to metabolize an adult diet.
Okay, so they were looking for a metabolic upgrade. Did they bring better tools to digest our food?
Yes. They dug into carbohydrate-active enzymes, the CC families, specifically analyzing the genomes of adult-derived versus infant-derived strains.
They were hunting for variations in carbohydrate utilization genes.
Right. They systematically analyzed 49 different CZ families, and from there, they selected 5 highly specific genes based on literature reviews to analyze at a very granular level.
Let's get into the weeds here. What were the specific genes?
We're talking about genes like GH36, which codes for RAFA,
okay,
and GH42, or BKB. There's also GH2, which is lanKC, GH20, which is NA, and finally GH112, or LNBP.
That is quite the alphabet soup. But basically, these are the genetic blueprints for the enzymes required to cleave complex sugars, right?
Precisely. And the standard expectation in microbiology is that the adult-derived strains would show a significantly different distribution or maybe a higher abundance of these specific genes.
Giving them a metabolic edge to just feast on an adult diet, like they out-eat the competition?
That was the thought. But the statistical analysis completely derailed that hypothesis.
Wait, really? Completely derailed it?
Completely. Across these core genome features and these specific carbohydrate genes, the result was NS, not significant.
Oh, wow. So there was no statistically significant difference between the two groups?
None at all. The adult strains and the infant strains possess the exact same metabolic toolkit.
So it's not about who brought the best snacks to the party?
No. It forced a total pivot in the research. The ability to colonize the adult gut isn't driven by what the bacteria eat. It's not some shared metabolic trait across the species.
So they had to accept that survival in this crowded environment was being driven by, what, highly specific hidden genetic traits?
Yes, traits localized to individual strains rather than a broad species-wide dietary adaptation.
Okay, this is a perfect time to pause. Let's do a quick recap, like a bulleted summary of what we've established in this first segment.
Sounds good.
Okay, bullet 1. B. breve is usually an infant microbe, but adult-derived strains do exist.
Right.
Bullet 2. We thought they survived by having special food-digesting genes for adult diets, but they don't.
Exactly.
And bullet 3. Since diet doesn't determine survival, the secret weapon has to be some unique, hidden genetic trait.
That's a perfect summary.
So if they aren't out eating the competition, they have to be out competing them in some other way. What is the actual mechanism keeping them alive?
Well, to find that mechanism, the researchers had to move away from the core genome, which are the genes shared by all B. breve, and analyze the accessory genome.
The accessory genome, okay.
Yeah.
And they focused heavily on one highly successful adult-derived strain, right?
Yes, a superstar strain they designated as B. breve CCFM 1025.
CCFM 1025, that's our successful colonizer. So they put its genome side by side with the infant strains to run a massive comparative analysis.
Exactly. They analyzed 930 orthologous gene groups. And through this comparative genomics, they isolated 34 unique genes present in CCFM 1025 that were entirely absent in the infant-derived strains.
34 unique genes? That's a needle in a haystack.
It is. But as they mapped the function of these unique genes, they zeroed in on a highly specific grouping called cluster 2.
Cluster 2, okay. What's special about it?
It was identified as a lanthapeptide 3 cluster. And right at the core of this cluster, they identified the main driver, a gene they named lanKC.
Here's where it gets really interesting, folks, because lanK is a bacteriocin gene.
Yes, it is.
And for a quick refresher, bacteriocins are essentially natural antibacterial weapons. They are toxic peptides that bacteria synthesize and secrete to kill off their competitors.
It is localized biological warfare. I mean, in a densely packed ecosystem like the adult gut, where space and nutrients are the ultimate limiting factors, you can't just passively exist.
You have to actively displace the current residents.
Exactly. And bacteriocins, like the one encoded by lanKC, are highly specialized molecules. Because it's a lanthapeptide, it contains thiourether bridges.
Thiourether bridges meaning it's really tough.
Very tough. It makes the peptide incredibly stable, even in the harsh, highly acidic environment of the gut.
So it survives the acid.
Right. And it allows the CCFM 1025 strain to effectively punch holes in the cell membranes of neighboring competing bacteria.
Oh, cruel.
Yeah, it disrupts their proton motive force and causes cell death.
Okay, I have a mnemonic to help you remember this. Think of lanKC as the local area network kill command.
Oh, I like that.
Right, LA and Hayseed local area network kill command. Think of the gut as a nightclub that is already at absolute maximum fire code capacity.
It's packed.
You can't just walk in and find a table. CCFM 1025 survives because it brings its own bouncer. The lanKC gene encodes a bouncer that physically removes competitors, clearing the necessary real estate so the bacteria can actually establish a colony.
That structural metaphor captures the dynamic perfectly. But, you know, in molecular biology, just identifying a gene that codes for a bacteriocin isn't enough.
You have to prove it actually works.
Exactly. You have definitively proved that the bacterium deploys it in a real-world scenario and that this single peptide is the sole variable determining survival.
Wait, I need to challenge this whole premise before we look at the proof.
Okay, go ahead.
If CCFM 1025 is armed with this highly stable lanthapeptide and it's punching holes in cell membranes left and right, why doesn't it just act like a broad-spectrum antibiotic?
That's a great question.
Like, how is it not just acting like a nuclear bomb in the microbiome and wrecking the host's gut?
And that right there is the defining difference between a bacteriocin and a traditional antibiotic. Antibiotics are generally broad-spectrum. They wipe out everything, good and bad.
Right, scorched earth.
But bacteriocins are narrow-spectrum. They're highly targeted. They usually only affect closely related bacterial species that are competing for the exact same ecological niche and the exact same nutrients.
Oh, so it's not killing everything.
No, the lanKC peptide binds to specific receptors on the membranes of those direct competitors. It's a sniper rifle, not a hand grenade.
Okay, that makes a lot more sense. It's targeting the specific bacteria sitting at the table at once. So how did the researchers actually prove this targeted assassination works?
They utilized gene knockout validation.
Okay, what does that involve?
They took the successful CCFM 1025 strain, and using genetic engineering, they precisely excised only the lanKC.
They took the weapon away.
Exactly. This created a mutant version, referred to in the paper as lanKC delta lanKC.
Delta usually meaning change or difference, so the one without lanKC.
Right. They then ran a head-to-head comparison between this unarmed mutant and the normal, fully armed wild-type strain.
They fired the bouncer. Now we have the wild type with its targeted weapon,
Right.
and the mutant without it. So what happened in the test?
Well, the first phase of testing was just establishing a baseline in a pure liquid medium. They cultured both strains in separate, uncrowded, nutrient-rich environments.
Just living the good life.
Yeah, plenty of space. They tracked the optical density, which is how we measure bacterial growth at 0, 6, 12, and 18 hours.
And the 18-hour mark is crucial here, right? Because it simulates the transit time through certain models of the human digestive tract.
Precisely. And over those 18 hours, the growth curves for the wild type and the unarmed mutant were perfectly identical.
Identical, so removing the weapon didn't hurt it at all.
Not in that environment. This is a vital control step.
Yeah.
It proves unequivocally that the lanKC gene has zero involvement in basic metabolic survival, eating, or reproduction.
Ah, I see. When space and nutrients are unlimited, the bacteria doesn't waste energy deploying the weapon.
Exactly. But, as we establish, the adult gut is the furthest thing from an unlimited, uncrowded environment.
It's the pack nightclub.
Right. Which brings us to the second phase of testing, in vitro fecal fermentation.
The messy part.
Very. The researchers gathered fecal matter from 10 different human donors.
10 distinct donors, so it's a good cross-section.
Yes, to create a highly diverse, intensely competitive, generalized model of the human microbiome. They introduced a 2% volume-to-volume inoculation of either the armed wild type or the unarmed mutant into this dense microbial soup.
Literally throwing them into a simulation of the exact environment sitting in your digestive tract right now.
Yes.
So how do they measure who is winning a turf war when there are thousands of different bacterial species interacting simultaneously? Like, how do you even see that?
They utilized shotgun metagenomics.
Okay, break that down for us.
So older sequencing methods, like 16S RNA sequencing, they only look at a single marker gene, which can sometimes only identify bacteria down to the genus or family level.
It's a bit blurry.
Right. But shotgun metagenomics sequences all the genomic DNA in the sample.
Yeah.
It literally shatters the DNA, sequences the fragments, and pieces them back together.
Oh, wow.
Yeah, it gives a high-resolution, incredibly precise picture of exactly which strains are present and their relative abundance over the 18-hour fermentation.
Okay, so they have this super clear picture. When they ran that high-resolution analysis, what did the data actually show?
The divergence was massive. Over the 18 hours, the relative abundance of the armed wild-type strains significantly increased.
It won the turf war.
It did. It successfully carved out a niche and established a foothold in that competitive environment.
And what about the mutant strain?
The mutant strain, the lanKC, without the bacteriocin gene, it flatlined. Its abundance absolutely plummeted.
Wow, really? So without that one gene?
Without that one specific lanthopeptide third cluster, it was completely outcompeted by the native microbiome.
Okay, time for our second quick recap before we move on to the network effects.
Let's do it.
Bullet 1. The adult-derived strain, CCFM 1025, possesses this unique bacteriocin weapon gene, lanKC.
Yes.
Bullet 2. The knockout experiment proved this gene isn't for digestion, it's purely for competition. They grew exactly the same when they were alone.
Exactly right.
And bullet 3. In a simulated, crowded human gut, that single gene is the literal difference between successfully colonizing and being completely eradicated.
That single gene dictates survival. But the researchers didn't stop at mere survival.
They wanted more.
They wanted to understand the structural impact on the entire microbiome. You see, when a new bacteria moves into a neighborhood and starts forcefully evicting residents, it changes the local ecology.
Right. It shakes things up. So how do you measure that?
To measure this, they utilized ecological network analysis.
This is where we stop looking at the gut as just a list of bacteria, right? And start looking at it as a complex web of interaction. Basically, graph theory applied to biology.
Exactly. You map the microbiome using nodes and edges. The nodes are the different bacterial species, and the edges are the interactions between them.
Okay, I'm picturing a big web.
Yeah, and the researchers calculated specific topological metrics, most notably two things: robustness random and cohesion.
Let's clearly define those metrics for the listener. What physically happens to the gut network when robustness random increases?
So robustness random measures the structural integrity of the microbial community when it faces random environmental shocks.
Like what kind of shocks?
Like if you introduce a sudden dietary shift or a course of antibiotics or a pathogen, does the community network shatter or does it hold its shape?
Ah, okay. And what did the data show?
The data showed that the armed wild-type strain significantly increased the overall robustness of the gut network compared to the mutant.
It fundamentally reinforced the neighborhood. It made it stronger.
Yes, and then there's cohesion.
Right. What is cohesion?
Cohesion measures the interconnectedness of the community, and it's divided into positive and negative cohesion.
Okay.
The wild type increased both. Now, negative cohesion often sounds detrimental, but in ecological network theory, it represents strong competitive exclusion.
Meaning what exactly?
It indicates that the network is highly efficient at repelling invasive, potentially pathogenic bacteria. It essentially tightens the zoning laws of the neighborhood.
So the lanKC gene doesn't make CCFM 1025 a neighborhood bully, it makes it a neighborhood watch captain.
I love that analogy.
Yeah. The bouncer isn't just protecting CCFM 1025, it's protecting the entire block by keeping bad actors out, increasing that negative cohesion.
Exactly. And that leads directly to the positive cohesion. The shotgun metagenomics data revealed that the armed wild type strain was positively correlated with several established beneficial species.
Oh, so it makes friends too. Which ones?
Specifically, a Metabacterium intestinale, Ruminococcus lactaris, and Bacterium hominis.
Okay, but why those specific bacteria? If the bacteriocin is clearing space and acting like a sniper, why did these specific beneficial species thrive alongside it instead of getting wiped out as collateral damage?
It goes back to the narrow spectrum nature of the lanKC bacteriocin we talked about. It selectively targets direct competitors for CCFM 125's specific metabolic niche. Ruminococcus lactaris, for example, occupies a completely different metabolic niche.
So they aren't competing for the same food.
Exactly. By assassinating its own direct competitors, CCFM 1010-5 inadvertently freed up physical space and secondary resources in the mucosal layer that allowed these other beneficial microbes to flourish.
It's an incredibly elegant system, but I have to play the skeptic again here.
Please do.
Petri dishes and in vitro fecal slurries, even with 10 donors, are still heavily controlled lab environments,
That's true.
The human digestive tract has an active immune system, a continuous flow of digestive acids, and constant dietary fluctuations. Does this localized biological warfare actually translate into a living human host?
Lab data always requires in vivo validation, and the researchers organized a human colonization trial to bridge that gap.
Oh, they actually tested it in people.
Yes. Participants were administered the armed wild-type CCFM 10205 strain. The researchers then tracked the composition of their microbiomes through fecal sampling at a 14-day mark and, crucially, at a 28-day washout mark.
The washout period is the real test.
Absolutely.
That means checking the JUT 28 days after the participants completely stopped taking the probiotic, just to see if the bacteria actually stayed and colonized, or if it had just washed out of the system.
Right. They categorized the human participants into two distinct groups based on the sequencing data. First, the colonizers, where the CCFM 1025 strain successfully engrafted and maintained its presence after the washout.
And the second group?
The non-colonizers, where the strain failed to establish a long-term presence.
Okay, and when they cross-referenced those human groups with the network analysis we just talked about?
The human data perfectly validated the in vitro simulations.
No way.
Yes. The participants in the colonizers group maintained a significantly higher overall proportion of the Bifidobacterium genus. Furthermore, when they ran the ecological network analysis on the human samples, the gut networks of the colonizers exhibited significantly higher robustness random and higher negative cohesion compared to the non-colonizers.
That's incredible. So the underlying mechanism operated in the human gut exactly as it did in the test tube.
Exactly. The lanKC bacteriocin structurally stabilized the human microbiome.
That is a phenomenal validation of the mechanism. But looking at the broader biological landscape, is CCFM 1025 just a freak anomaly? Like, did this one specific strain of B. breve just get incredibly lucky with a random mutation?
Well, biology rarely relies on a single isolated fluke, especially when a mechanism is this effective.
Fair point.
To contextualize this, the researchers applied an evolutionary perspective using large-scale phylogenetic tracing.
Tracing its family tree, basically.
Exactly. They queried massive genomic databases, specifically the Genome Taxonomy Database and the Early Life Gut Genomes, or ELGG database, which houses over 32,000 distinct genomes.
32,000. So they were looking to see if other species had developed the same bouncer, so to speak.
Precisely. They searched for the exact lanKC gene sequence across the entire bacterial phylogenetic tree, and they found that potential homologs, meaning genes with shared ancestry and function of the lanKC 1025 gene, are widely distributed across various species.
Oh, really? Who else has it?
They identified it in B. pseudocatenulatum, B. casuanohense, B. bifidum, and B. longum.
Which indicates this is a deeply conserved evolutionary trait.
It proves that this specific lanthopeptide bacteriocin strategy is a shared, highly successful evolutionary advantage.
It's not just a fluke.
Far from it. Over millions of years of microbial evolution, disparate species within the gut have either laterally transferred or retained this specific genetic weapon because it remains the most metabolically efficient way to guarantee ecological adaptation and long-term survival in the hostile environment of the adult gut.
Okay, let's do our final 5-minute mini recap to pull all this together.
Go for it.
Bullet 1, the lanKC gene doesn't just help the bacteria survive, it increases community stability and robustness.
Yes, the neighborhood watch.
Exactly. Bullet 2, it actually helps other good bacteria thrive alongside it by selectively taking out the competition.
Right.
Bullet 3, human trials prove it allows for successful long-term colonization, surviving that 28-day washout.
A huge finding.
And bullet 4, this isn't just one lucky bug, it's an evolutionary survival tactic shared across multiple gut species.
That's the perfect synthesis.
So, bringing all these threads together, what does this all mean? We started with a fundamental ecological mystery, how a microbe normally restricted to infants manages to survive the adult gut.
Right, a huge paradox.
We discovered that survival has absolutely nothing to do with superior digestion or a specialized diet. Instead, the comparative genomics isolated the lanKC bacteriocin gene,
A bouncer.
the bouncer, and through the knockout validation, we saw how that single gene acts as a highly targeted, narrow-spectrum weapon. We watched the network analysis prove that this localized warfare doesn't destroy the gut, but actually increases structural robustness and invites other beneficial species to thrive.
It builds a better city.
And finally, the 28-day human washout trials and the 32,000 genome phylogenetic trace proved that this mechanism is both highly effective in living hosts and a cornerstone of microbial evolution.
You know, when you look at the macro implications of this paper, it fundamentally disrupts the current paradigm of probiotic design.
How so?
Well, for decades, the industry standard has been a numbers game formulating probiotics with tens of billions of CFUs, multiple generalized strains, and basically just hoping something sticks.
Just throwing spaghetti at the wall.
Exactly. But this research demonstrates that sheer volume is completely irrelevant if the bacteria lacks the specific genetic architecture to compete.
If it doesn't have the bouncer, it doesn't matter how many you send in.
Right. It's a single gene, a minuscule sequence of nucleotides, dictates whether a probiotic successfully engrafts or is simply expelled. The future of microbiome therapeutics has to be precision engineering.
Wow, that's a huge shift.
We are moving rapidly toward an era of personalized bacteriology, where we analyze a patient's specific ecological network and prescribe a strain armed with the exact bacteriocin profile required to carve out a niche in their unique microbial city.
That is wild. The next time you sit down for a meal or, the next time you hold a bottle of probiotics, I really want you to think about the immense, invisible turf war happening inside your digestive tract at that very moment.
It never stops.
Does the bacteria you're introducing actually have the genetic tools, the biological weaponry to navigate that microscopic city? Or is it just showing up to an overbooked, highly exclusive environment without a way to get through the door?
It's a fascinating thought.
It really is. The microbiome isn't just about what bacteria eat, it's about how they fight. And understanding that completely changes how we view human health. We'll leave you to ponder the microscopic battles happening inside you right now. Thanks for joining us on this deep dive.